Bonjour à toutes et tous. On va commencer. Donc c'est le premier séminaire de l'année, on commence en anglais. C'est peut-être le début d'une longue série. Donc je suis... it's French. Donc je suis très contente de présenter Emma Anderson. Emma is a postdoc with MRC fellowship. She was trained at Bristol University in Wales. That was Europe. That's too bad. Wow. Oh yeah, the other side is Wales. Yeah, I'm sorry. Okay, but so the rest of my sentence is true that you're in Europe still. Emma is visiting us for a month as part of postdoc training, and she is going to speak about education and dementia.
So thanks to Carole for inviting me to give a talk. As she said, this body of work is part of my postdoctoral training. So I was just awarded a 3-year fellowship to improve causal inference in studies assessing risk factors for dementia. So this work that I'm presenting today is kind of the first step. It's the first bit of work that I've done in my fellowship. Can you turn the mic up? The mic?
Yeah.
Is it okay? Okay. So the observational evidence that we have at the moment for the association between educational attainment and Alzheimer's is generally quite consistent. So it always tends to show a protective effect of education on Alzheimer's. But there is still some debate as to whether this causal effect is actually representing a true biological effect, or whether it's maybe confounded by factors like social class, or like so other lifestyle risk factors that are associated with both educational attainment and Alzheimer's. So some studies have tried to get around this problem of potential confounding by using a technique called Mendelian randomization to examine this specific question. So I'm not sure how familiar everybody here is with Mendelian randomization, but I will just give some information just to kind of bring everybody that doesn't know up to date, because otherwise the rest of my talk will make absolutely no sense. So Mendelian randomization is a form of instrumental variable analysis. So it's where we use a genetic instrument to proxy for an environmental exposure. So Z is the genetic instrument and X is the environmental exposure. So when we do this, we're able to estimate the causal effect of X on Y. And the reason that we're able to do this is because your genes are randomly allocated at conception. And because of this, both observed and unobserved confounders are likely to be evenly distributed among the genotypes. So when we do Mendelian randomization, we take the exposure, the association between the gene and the exposure over the association between the gene on the outcome. And this is generally done in one sample. So the gene exposure and the gene outcome association is obtained from the same sample. So when we do Mendelian randomization, there are a few assumptions that have to be met for it to be a valid test. So for us to be able to make causal inference, there are some assumptions that have to be met. So the first one is that the gene has to be, sorry, I keep losing my, the gene has to be robustly associated with the exposure. The second assumption is that Z is, there is no arrow from Z to U or from U to Z. So there has to be no association between the genetic instrument and the confounders. And the final assumption is that Z can only influence the outcome via the exposure. So there can't be a direct effect of Z on Y, or another indirect effect of Z on Y that doesn't go through X. So those are the assumptions that we need to meet to do a Mendelian randomization study. So There are two Mendelian randomization studies that have looked at whether educational attainment is causally related to Alzheimer's disease, both of which used an old genome-wide association study for educational attainment. So they only had 3 SNPs to proxy for educational attainment, and these explained approximately 2% of the variance in years of schooling, which was the unit that they measured educational attainment by. So the evidence from these 2 Mendelian randomization studies are conflicting. So one showed a protective effect of education, as we might expect from the observational literature. And the other one, which is showed here, the more recent one, showed a null effect. So there was no effect of education on Alzheimer's. So this is potentially contradictive of some other literature that we have from different forms of instrumental variable analyses, where instead of using a genetic variant to proxy for education, we use things like policy change. So as an example, in the UK, we recently had a rising of the minimum age at which you could leave school. So these policy changes can also be used to proxy for educational attainment, and in those studies where we use this policy change, we again see a protective effect of education on Alzheimer's. So it could just be that these Mendelian randomization studies are underpowered. So because they're only using 3 SNPs and they're explaining such a small proportion of the variance in years of schooling, it might just be that we don't have enough power. And the other important point to make is that the 2 existing Mendelian randomization studies, they did no sensitivity analyses to check how robust the causal effect estimate was to the various assumptions that we make in Mendelian randomization. So that really limits our ability to make causal inferences from these 2 studies. So last year, a new genome-wide association study was published for educational attainment, and they identified 74 independent loci that were associated with years of schooling. So these 74 loci explain about 3.5% of variance in years of schooling, so it's not a huge improvement on the 2%, but we do have many more SNPs. So the aim of my particular study was to kind of redo this Mendelian randomization study using this larger SNP list, which gives us much more power, but importantly to also conduct a series of sensitivity analyses that check how robust our estimates are to the various assumptions that we make in Mendelian randomization. And as a secondary aim, what we then did was to see whether our effect estimates were potentially biased by survival, so whether there was any frailty effect. So we actually conducted a 2-sample Mendelian randomization. So when I explained Mendelian randomization just now, I said that the gene exposure and the gene outcome association is generally estimated within the same sample. That can be tricky, particularly for something like Alzheimer's, where we don't have very, very large studies with the numbers that are needed to conduct an MR analysis. So a 2-sample Mendelian randomization is an extension of MR, where the relationship between the gene and the exposure and the relationship between the gene and the outcome are obtained from 2 different studies. So I've tried to just demonstrate this here. So in your exposure GWAS, you would have the effect sizes of each SNP on your exposure. You would then look up those SNPs in the GWAS of the outcome, and that gives you the association between the genes and the outcome. So then the causal effects are combined using an inverse variance weighted approach. So that gives you an overall effect estimate of educational attainment on Alzheimer's. And I should say that we actually use 63 SNPs from the discovery sample of the genome-wide association study. This was just after removing palindromic SNPs and after harmonization, so we actually ended up with 63. So this is the only French that you'll get through my whole talk. So as I said, one of the aims was to conduct a series of sensitivity analyses. So one of the most important ones that we looked at was a method called MR-EGR. So when you use lots of alleles in an MR analysis, you greatly increase the potential to observe pleiotropic effects. So pleiotropy is where you have a single gene and it acts on multiple different pathways. So when we do an MR-EGR regression, the intercept gives you the overall pleiotropic effect of the SNPs on the outcome. So this is the effect, it's the direct effect of the SNPs on the outcome that don't go via the exposure, which invalidates one of the assumptions of MR. And the beta coefficient that you get from an MR-EGR, or the slope, is the causal effect estimate of education on Alzheimer's that is adjusted for this level of pleiotropy. So the MR-EGR estimate is only consistent with your main IVW estimate, the inverse variance weighted approach, if there is no pleiotropy present. And when you do this method, so when you conduct an MR-EGR analysis, you make the assumption that there is no measurement error in your gene exposure association. So one of the guys in Bristol, Jack Bowden, he developed a method which is an extension of what we use in meta-analyses. It's a measure of heterogeneity. So he's developed a statistic called I²GX, and this provides an estimate of the degree of regression dilution in the MR-EGR causal effect estimate that's due to the uncertainty in the SNP exposure association. So when you have this estimate, you can then do a simulation extrapolation to adjust your MR-EGR causal effect estimate for this degree of regression dilution that's due to this measurement error. We also conducted a weighted median analyses. So this accounts for potentially invalid instruments, genetic instruments. So the weighted median will only give you a consistent estimate of the causal effect, so consistent with the IVW, if at least 50% of your weights come from valid genetic instruments. And by valid, I mean robustly associated with the exposure, not associated with confounders, and the effect goes via the exposure of interest. This is a bit of a large table, but it's basically just to summarize kind of one set of sensitivity analyses that we did. So as I previously mentioned, the IVW, MR-EGR, and weighted median analyses that we did were conducted on SNPs that were identified in the discovery sample of the genome-wide association study. So we conducted a series of sensitivity analyses to check whether our results were sensitive to Winner's curse. So Winner's curse is where the effect sizes of variance identified within a single sample are likely to be larger than they are in the overall population, even if there is a true, even if there is a true association between the gene and the exposure. So we checked this by repeating our analyses. I show in this table only the IVW, but we did this also for weighted median and for MR-EGR. So we checked it by repeating analyses using the 63 SNPs obtained in the replication sample of the GWAS. So that was actually UK Biobank. That was the replication sample that they used. We then looked at a set of SNPs that were identified in the meta-analysis of both the discovery and the replication sample. And then finally, we looked at the set of SNPs that were present in the meta-analyses, so this 128 SNPs, minus those that were in the discovery samples. So this is a totally independent set of SNPs. So it is subject to Winner's curse again, but it is sort of, independent evidence for causality. I should also say that we did some leave one out permutations, which I'll show you in the results, but essentially this assesses, again, a pleiotropic effect of single SNPs on your causal estimates. So one of the aims was to look at whether there was potential for survival bias in these causal effect estimates. So if both an exposure and an outcome are associated with mortality, the estimate of the effect of the exposure on the outcome can be biased, even in Mendelian randomization studies. So for example, if people with lower education are more likely to die before the onset of Alzheimer's, then bias can occur because those with a genetic predisposition to higher educational attainment are more likely to live longer, and so by definition, they're more likely to receive an Alzheimer's diagnosis. So we did this by performing simulations, and in our simulations we assumed, or we stipulated, that there was no effect of education on Alzheimer's. So we were then able to estimate the level that the level of the causal effect estimate that we would see that was due to survival bias alone. So if we observe any effect in those analyses, this is due to survival bias and not a true causal effect of education on Alzheimer's. So we did this by conducting 1,000 simulations on 500,000 individuals. So everybody in the simulation study is allocated a genotype for the 63 SNPs, and that's based on the allele frequencies within our own MR analyses. For each individual, years of schooling were simulated as a function of their genotype and the effect sizes for the SNPs, plus a random value to create a population variance of 1, and that's just because we were using standard deviations of years of schooling. An age variable was generated for everybody to match the distribution of ages that were in the Alzheimer's GWAS, so the age distribution among the cases and controls. Mortality status was computed as a function of age and also education. So we used the Gompertz-Macomb mortality curve, and everybody had their alive or dead status sampled as a function of the probability of death due to this curve. And then Alzheimer's status was simulated as a function of age-related diagnosis rates. So Alzheimer's was a function of age and age only. There was no causal effect of education on age. So once we had all of our people, there was a subset of individuals both with and without Alzheimer's who were retained. And this was based on the distribution of ages within the Alzheimer's GWAS. So then we conducted our MR analyses on the simulated data set and also observational analyses. So this enabled us to gauge the extent to which any association between education and Alzheimer's might be artificially induced by frailty effects. So here are the results. Oops. So this table here shows the causal effect estimates from the IVW, the weighted median, and the MR-EGR. This is the effect of each SNP, and this is the slope for each of the regression methods here. So the main IVW regression using all of the SNPs from the discovery sample of the education GWAS showed that with each standard deviation increase in years of schooling, so that translates to about 3.5 years of schooling, the odds of Alzheimer's were on average reduced by around 37%. The MR-EGR regression showed little evidence of pleiotropy. So this is the intercept estimate here, and there's no evidence that this differs from 0, so there's no evidence of a pleiotropic effect. The causal effect estimate from MR-EGR was generally similar to the main IVW effect, but the standard deviation was much, much larger, and this is expected. So in MR-EGR, we're estimating 2 parameters, both the intercept and the slope, whereas in the IVW, you're only estimating the slope, so you always end up with larger standard errors. The weighted median estimate was also similar to the IVW. Again, the standard error is larger, and this is mainly just due to the fact that you're using only 50% of the weights from your genetic instruments. Oops. So this was the result from the I²GX. So 0.67 suggests around a 33% attenuation in the causal effect estimate, the MR-EGR causal effect estimate. And when we did the simulation extrapolation, that gave us a corrected MR-EGR estimate of 0.63, which is entirely consistent with what we see in the IVW. This is the graph that I was talking about with the leave one out permutation. So if any one of these SNPs were invalid, then you'd expect to see some distortion in the distribution of these results, but they're all fairly well aligned. So that, again, is no evidence of pleiotropic SNPs. This is the sensitivity results for the main IVW analysis, but they were similar also for MR-EGR and weighted median. So this is checking susceptibility to winner's curse. The causal effect estimates were slightly larger when we used the SNP effect estimates that were derived from the replication sample in UK Biobank. And this is entirely consistent with a dilution of the causal effect estimate when you use SNPs from a discovery sample. So it's exactly what we would expect to see. It shows that we do essentially have some winner's curse in our main effect estimates. This is the simulated mortality curve. So this is just to show that individuals with higher levels of educational attainment are slightly more at risk of being diagnosed with Alzheimer's due to their increased longevity. And these are the results from the simulation analyses. So this table shows the mean and standard error of the effect estimates over the 1,000 simulations that are due to selection bias. So you can see that there is some evidence that there is an effect induced due to survival in the observational associations, but that there wasn't any survival effect in the IVW or the MR-EGR. So to conclude, our IVW suggests that per around 3.5 years of additional schooling, the odds of Alzheimer's are on average lower by 37%. The weighted median and the MR-EGR sensitivity analyses were generally consistent with the IVW, and the results were robust to the removal of each SNP in turn in the leave one out permutations. So there was no evidence of pleiotropy. And the corrected MR-EGR estimate was very similar to the IVW, which again is evidence that there is no pleiotropy. So overall, our findings support a causal role of educational attainment on Alzheimer's. There is some further work that's planned, so we would like to, if possible, assess whether the magnitude of effect for educational attainment on Alzheimer's is different, basically in different types of education. So is this effect similar in people that complete academic versus vocational training? Is it the same for people that study part-time compared to full-time? And does this effect change if you then if you are educated more in later life? So say if you don't have your schooling earlier in life, but you then later go on to attend a night school, does this effect remain consistent? We're also putting in a grant to look at potential mediation. So we'd like to do some network Mendelian randomization analysis, just to try to unpick the pathway and see why education is associated with Alzheimer's. So is it because it affects different lifestyle characteristics? Does it make you less likely to be obese, less likely to smoke, which then subsequently affects your Alzheimer's risk? Or is it due to some biological effect, maybe by increasing brain volume or hippocampal volume? So that's kind of the work that we've got planned to take this further. And these are my acknowledgements. This is just my co-authors in Bristol. Jack Bowden, he's developed most of the methods that I've used in this presentation. So if anyone's interested in doing Mendelian randomization studies, he'd be a great person to contact. He's really lovely, and drop him an email because he's fab and he can help you out. Oh, and the reference is here for the paper's written up and is on bioRxiv. It's currently with BMJ. So yeah, if you want to read the paper, that's the reference. I'm happy to take any questions.